first question is from GATE 2017. So here they ask that the molecular weight of a protein is determined by a native page, which native page is 400 kilodalton. This protein, when run on a non-reducing SDS page, gave a band of 200 kilodalton, and on a reducing SDS page, it gave a band of 100 kilodalton. So, how is the protein? So, the options are four subunits, of which two subsets are linked by two disulfide bridges. Option B, four subunits which are linked by four disulfide bridges. Option C, two subunits only and none are linked by disulfide bridges. Option D, two subunits which are linked by disulfide bridges. So, what would be the answer? First of all, the native in the native page, the actual protein weight will be shown by 400 kilodalton. I hope you all know about the differences between native page and SDS page and why SDS is added uh, to the protein, because that basic understanding is required to answer these questions. So, in a native page, protein is separated in its native form, like all its if even if it is a oligomer or a monomer, it does not matter. It will run in its native form. So we will get the actual molecular weight of the protein. So that is 400 kilodalton. So we do not know from here. We do not know whether the protein is like it has various oligomers or it is linked by disulfide bridges or not. We cannot know. It gives only the native size. This protein, when run on a non-reducing SDS page, when it is run on a non-reducing SDS page, that means the SDS page is there. SDS is there, that is the protein will get denatured, but it is a non-reducing page, that is beta mercaptoethanol or DTP is not added. So the disulfide bonds between the protein are not broken. So if the protein is run on that, then we get a band of 200 kilodalton. So we get 200 kilodalton. Earlier we were getting 400 kilodalton in native page, then we get 200 kilodalton bands. Then on and when we ran a reducing SDS page, okay, the disulfide bridges are broken here. It gave a band of 100 kilodalton. So on SDS page, this protein further gave a band of 100 kilodalton. So how can we determine what is the structure of the protein? So basically the protein has the basic monomer of the protein is composed of 100 kilodalton and the total size of the protein is 400. So there should be 4 of these 100 kilodalton monomers. So there are 4 subunits. So it can be option A or B. Now how to determine how they are linked? Now you see in a non-reducing SDS page, it gave a band of 200 kilodalton. That is, there are two disulfide bridges that will, so you get, so each of these 100 kilodalton monomers are joined to each other by a disulfide bond. Okay, these two are again joined to each other by a disulfide bond. That is why on a SDS page, on a non-reducing SDS page, they migrate together as a 200 kilodalton because the disulfide bond is non. So this is 200 kilodalton. This one, this is 100 kilodalton. That is linked together by a disulfide bond. So in a total, this 400 kilodalton protein, there are four monomers, okay, and two sets are linked by disulfide bridges. So over here, these two may be linked with disulfide bridges, and these two are linked with disulfide bridges. This is the answer. Answer is A.